this video, we're going to talk about the single-stranded RNA virus influenza, which is the cause of the flu. We'll talk some about the structure of the viral particle and some of the proteins that are encoded by influenza, and then we'll discuss the attachment, entry, and uncoating of the virus, as well as its biosynthesis, and how the unique structure and the polymerase of influenza allow for its evolution. The viral particle of influenza is enveloped and spherical. Recall that enveloped refers to the fact that this contains a phospholipid bilayer around the capsid protein. That phospholipid bilayer comes from the previous host that the virus infected. The genome of influenza is segmented. You'll notice in the middle of this image here that there are 9 different genomic segments for the influenza genome. And this genome here is made up of negative single-stranded RNA. There are a number of different proteins that are required for influenza to cause an infection. First, the proteins on the outermost part of the envelope, hemagglutinin and neuraminidase, are required for attachment and exit, respectively. The M1 matrix protein makes up what we would consider to be the viral capsid, while the M2 protein is a hydrogen ion channel, and this will be required for uncoating. Shown here is the nucleoprotein. This is bound to the RNA. Not only does it protect it, but it helps to package that RNA during the assembly of the virus. And then finally, we have 3 polymerase subunits, which together make up an RNA-dependent RNA polymerase. Influenza infects epithelial cells. In the first step of infection, what we find is that the hemagglutinin spike protein that is on the surface of the viral particle attaches to sialic acid. This then triggers penetration by receptor-mediated endocytosis. Within what we call the endolysosome, what we find is a reduction in pH, and this is going to induce the uncoating of the viral particle. The viral genome and its associated nucleoproteins will localize to the nucleus where they will begin biosynthesis. We find that the synthesis of viral genomes occurs, mRNA is made, and then viral proteins will be synthesized on ribosomes outside the nucleus. These proteins may be modified within the Golgi apparatus. Next, we find that during the assembly of the virus, the viral genomes and their associated nucleoproteins are going to begin to interact at the cell membrane. The nucleoprotein will interact with the matrix protein M1, and M1 will interact with M2. This creates essentially a scaffold of proteins that are going to help to recruit each other to provide the self-assembly that we find within all viruses. In the last step, during the exiting of the virus, what we find is that influenza actually buds out of the host cells. And the envelope protein neuraminidase, or NA, is required to cleave sialic acid so that the viral particle can be released from the host cell. Let's talk for a couple of minutes about how this virus performs biosynthesis. First, remember that this virus has a negative single-stranded RNA genome. This type of genome is complementary and anti-parallel to mRNA. And because it has an RNA genome, this virus is going to have to encode its own polymerase. In the case of replication, what we're going to find is that this virus first will take each of its segments of its genome and convert them into plus single-stranded RNA. These are going to be RNAs that are the same length as the negative single-stranded RNA. And this will be performed with the RNA-dependent RNA polymerase encoded for by the virus. This plus single-stranded RNA then serves as a template to make lots of copies of that negative single-stranded RNA, and this is the replication of the virus. Now, in terms of transcription, the negative single-stranded RNA serves as a template to make mRNA. And so in transcription, this is going to require an RNA-dependent RNA polymerase again to make that mRNA. And all mRNAs in eukaryotes have two basic features. One of them is a poly-A tail, which I'm showing here at the end of this mRNA sequence. This would inherently be encoded for by the negative single-stranded RNA. The other thing that all eukaryotic mRNAs have are a 5' methylated cap. However, the RNA-dependent RNA polymerase and the machinery that's encoded for by influenza does not encode for a protein that makes a cap. And so instead, influenza has this really cool technique which has been called cap snatching, in which it essentially steals or decapitates the mRNAs of its host, and it takes that cap and it attaches it on to the viral mRNAs. This viral mRNA then, after processing, can be exported from the nucleus, and it will be translated by the host ribosome. Now, there's a couple of things that really do come from this cap snatching beyond just making the mature viral mRNA. First, it allows for that viral mRNA to be recognized by the host. Secondly, the host mRNA in response to losing that cap will actually be degraded. This provides nucleotides to drive the synthesis of viral mRNAs. And finally, because the host mRNAs are being degraded, it'll actually reduce the translation of those host proteins. That makes more ribosomes accessible to synthesize viral proteins. So just to recap the life cycle, let's take a look at this process one more time. We've talked about how the hemagglutinin on influenza will bind to sialic acid and will trigger receptor-mediated endocytosis. Fusion of endosomes with lysosomes will cause acidification. Hemagglutinin and the matrix protein will change shape, allowing for the virus to uncoat. Within the nucleus, we find the segments of the viral genome to undergo replication. We also find that the viral genome will produce mRNAs, and those mRNAs steal the 5' cap from its host's mRNAs. The viral mRNAs are then translated, and in the processes that we have not talked about in as much great detail, we find that those proteins can be modified in the Golgi, and then the virus is going to exit from the cell by the process of budding. As the virus is budding, the neuraminidase, or NA protein, is going to be involved in cleaving the interaction between hemagglutinin and sialic acid to allow for these viral particles to then infect new host cells. Let's finish off our conversation on influenza by discussing how this particular virus evolves. There are two mechanisms at play that drive influenza's evolution. First are mutations that are generated during replication. Influenza encodes its own polymerase, and this RNA-dependent RNA polymerase has a rate of mutation roughly equivalent to 1 mutation for every 10,000 bases. This is similar to the mutation rate of other RNA viruses. And this leads to a process that we call genetic shift, where there are small changes in the genome of influenza over time. The second mechanism of evolution is more dramatic, and we call this genetic drift. This is what keeps epidemiologists up at night. Let's imagine the following scenario where strain A is an avian influenza and strain B is a human-based influenza. And you can see the colors of their genomes in green and blue. One organism that can be infected by both avian and human influenza viral particles is the pig or other swine. Let's imagine that both strain A and strain B infect a pig at the same time. If these two strains were to infect the same cell, what we might find is a reassortment of viral genomes from strain A and strain B during the infection. In this case here, that reassortment would produce a viral particle that would contain some genome from strain A and some from strain B. These very large wholesale changes are what have led to some of the biggest pandemics of influenza that our world has seen. So to summarize, based on the high mutation rate by the viral RNA-dependent RNA polymerase, and based on the fact that this virus contains a segmented genome, we can find that influenza will evolve at sometimes faster rates than other RNA viruses.